I think you guys all know this. What's the largest organ in the body? Skin. Oh yeah. And how much money do we spend globally per year on skin?
In this family?
Glasses, anybody? So we're here to hear about OneSkin Technologies. Claire has done a great job, and it's a pleasure to introduce Carolina and Alessandra, who are co-founders, the CEO and the Chief Scientific Officer. I'm gonna, the bios are all here in detail. But I want to hear the origin story first and foremost. That's sort of the exciting. And what was the moment in time when you said, "Okay, there's a better way to do this"? So why don't you come, take the stage? Thank you for coming out. We're grateful. I know you were going to host us at your place, but this turned out to be super easy, convenient, and you brought the samples with you anyway.
Yes.
Awesome. All right. Beautiful.
Hello, it's working, right? Okay.
Let's make that here.
Yeah.
Yes.
So thanks again, Peter. Thanks, Claire. It's a great pleasure to be here today. We're going to share a little bit about our story, about what we are doing at OneSkin, and we're going to do more in a conversation style. So if you have questions, feel free to interrupt us. We brought some samples. We can show a little bit how we do the testing in the lab, and we brought the skins that we grow. So you guys are welcome to come by and check it later too. So yeah, a little bit about myself. I have a PhD in stem cell biology and tissue engineering. I met Alessandra in the same lab in Brazil. We were doing master and PhD together. And when I was finishing my PhD in 2014, we were thinking how we could translate the science of stem cell biology to bring this to people. And we know the potential applications of stem cells to cure several diseases, but at that time, we thought that we could use the stem cells to produce human cells and offer this as a model so the industry would test their drugs and the cosmetics instead of using animals. So it was an alternative model for animal testing. We were inspired in this company here in the US, Cellular Dynamics. So basically they produce cells from the heart, cardiomyocytes, cells from the retina. I did my PhD producing cells from the retina to treat macular degeneration. And eventually we saw that, at least in Brazil, the demand for this kind of models that are more accurate, they are more representative of the human physiology than animals. The industry, the demand was not so big in Brazil, but the cosmetic industry was very much in need of these alternative methods, and growing human skin was the model that was already used in Europe for several years. And we decided to focus more on the skin and not only producing cells, but also growing human tissues in the lab. And eventually we came here to the US in 2016. We joined IndieBio, that's a biotech accelerator. So basically IndieBio also helps scientists to become entrepreneurs. And here was very interesting because we opened a lot our horizon, our vision, what we could do with that technology. So we started like seeing the industry, and we saw that this market that they call anti-aging market is a market that's only growing. It's only going to grow because people are only getting older. But we still don't know which products work and which products don't work, right? So we saw like a huge need for a validation, how we could develop a methodology to prove the efficacy of those products. And since we are able to grow tissues in the lab, if we're able to measure the age of the skin, and we saw that we could do this by the DNA. So I don't know if you have heard of molecular clocks, but basically our DNA accumulates epigenetic chains over time. And if we sequence our DNA, we're able to estimate our biological age. And then we saw that we could use this approach to test new products. So let's say that we are growing skin that's like 40 years old. And once we test with a product, if the skin is 37, then we can show that the skin has been rejuvenated by 3 years. So this would be a more quantitative way and a more objective way to validate those products in the market. And we start getting tractions. We saw some companies interested in having us testing their products. But when we start testing some products, we realized that none of them were targeting aging. They were trying to make your skin to look better, but there was no rationale behind that product. So we saw there an opportunity. Okay, there is a huge industry, there is a demand, and we saw the advances in the longevity science. So there is an opportunity to develop something that's really targeting the cause of skin aging, and then develop something that's more effective. So that's when we got very into the longevity community, and we saw the rise of cellular senescence, and then we decided to combine these two words, apply target cellular senescence as a way to prevent or to promote skin age reversal, and then eventually bring this as a new product that could actually really rejuvenate the skin in this molecular level. So that's a little bit the story, how we got to this. And what we want to bring here today is that skin is not, I know that everyone is still, the first thing that comes to our mind is that the skin is about beauty and aesthetics. But as Peter said, skin is our largest organ, and it has a huge influence in our overall health. So we want to bring this perspective of looking at your skin as an organ that's a part of your body that needs to be taken care of, and it plays an important role in regards to your longevity. So if you think that, I don't know if you heard of this word inflammaging, probably most of you have heard. So inflammaging is a process that your immune system has been chronically activated, so the levels of inflammation in your body goes up, and it's related to several age-related disease and chronic disease. And one of the sources of this inflammation could potentially be your skin, because again, skin being your largest organ, your skin deteriorating, accumulating senescent cells, those secretions of those senescent cells could be driving your body's inflammation. So we strongly believe in that if you take care of your skin, or if you treat your skin, or if you can, and this is a clinical study that we are planning to do, so if you treat your whole body with a product that was designed to decrease inflammation or target senescent cells, we can measure the levels of inflammation in your blood and hopefully see a decrease. So showing this connection and showing how skin could potentially help you to prevent diseases like Alzheimer's or cardiovascular disease. So just changing a little bit how we look at our skin, not because mainly for guys, right? Guys don't care much about like how I look, but if it's about health, I imagine that everyone is gonna take like 5 minutes and keep your skin hydrated, and even better if you can be targeting, the damage of your skin in the cellular level. So I'm sure that you guys are all familiar with senescent cells and the accumulation of senescent cells in different tissues. associated with several age-related disease, and skin is no different. These senescent cells, they also accumulate in our skin, and they are related with signs of aging, such as wrinkles, sagging, and even skin cancer. So targeting senescent cells is an interesting approach to promote skin health. Very quickly, we like to compare senescent cells with a bad apple because the main, I would say, problem of senescent cells is the secretion that they start producing in the tissue. So they start secreting inflammatory molecules that contaminate cells around, and this leads to inflammation, leads to tissue dysfunction. So by eliminating or preventing the formation of senescent cells, we can keep your tissue in a better health. And very similar to what we have in our healthcare system today, most of the interventions that we have available for skin rejuvenation, they are treating the symptoms of aging, right? Not the root cause. And ironically, a lot of those interventions, such as acids, they can be damaging your skin and accelerating aging in the long term. So we have seen a lot of treatments that when we incubate our cells, the markers of inflammations and aging go super high. And these are the molecules that are being used in several products that we are using every day. So we are fundamentally different in the way that we come from this longevity angle and trying to understand the biology of aging first. And then once we have a target, a new molecule that affects that target, we validate in the skins that we grow in the lab. And once we can prove the benefits that that molecule is promoting, then we bring this molecule to the market. So yeah, we have this different perspective. And again, by treating the cause, the consequence is that your skin will be healthier in the first place. And as a byproduct, your skin will also look better because it will bring your skin to a younger state. So beauty is still a consequence of like intervening in the root cause of skin aging. So for those who hasn't seen a skin in the lab, so this is what we brought here today. So we grow the skin. So this is like a ring of like gel. This gel is collagen that we mix with fibroblasts. That's the main cell of the derms. And then we add keratinocytes, that's the other cell of the epidermis. And we bring this construct to this interface air liquid. So the bottom of the skin will be in contact with the liquid that has nutrients. And the air will be the stimulus that will make the skin to differentiate and to form the same structure or a very similar structure that our human skin has. And then, so yeah, so this is a little bit of the protocol in terms of, mixing fibroblasts with this collagen, and then we add the keratinocytes here on top, and then we bring this construct to this interface, and the skin will differentiate, and in the end you have something like this. So this is a normal human skin. If I take a biopsy of one of you today, we'll see something similar, and this is the skin that we grow. So we can see then that we can recapitulate the main layers of this skin. The dermis here that contains collagen, fibroblasts, and brings this firmness to the skin, and then the epidermis with the stratum corneum that forms this the most important barrier to protect us against infections, water loss, and so on. So we can recapitulate these, and then we can test molecules. both by adding molecules in the media, in that liquid that the skin is in contact with, so we can test specific molecules. Whatever molecules you have heard in terms of has a potential to promote rejuvenation or target senescent cells, we have tested. Or we can also apply topical products in the top of the skin. But only grow human skin is not something that we created. Other companies do that. Our differential is to be able to replicate the skin aging process in the lab. So most of the companies that are offering skin tissues today, they are growing neonatal skin because neonatal cells, they replicate super fast. It's very easy to grow. But then they are treating anti-aging products in neonatal skin. That doesn't make sense, right? They don't need to be younger. So what we do, we use cells derived from donors of diverse ages. We have cells like from 0 to 30s, 40s, 50s, until 90 years old. And then we can replicate this aging process, and we can see how much the skin changes based on the cell that you use to grow. So the epidermis gets thinner, the epidermis, this purple layer gets thinner. Several aspects here change. We see the...
How many different collagen molecules are there? There's like, are genome codes for like, it's like 22 different collagen molecules? Is that the number? Do you know?
Yeah. On our 3D models now, we only like to start the first part to build the gel, to have the structure. We only use one collagen, collagen type 1, that is the most produced. But during the time like that the skin is growing, that takes 7 days, the fibroblasts that you add there, they start secreting all the other collagens that form the extracellular matrix. But although it's very similar to our human skin, our 3D skin is not complete with all the collagens that you have on a human skin.
One of the things I remember, and I was looking forward to checking it with you, was one of the reasons skin ages is the dysregulation that we stop manufacturing all the different collagen molecules. So we like shut down like 2/3 of them. Is there some truth in there?
Yeah, we can see, like, let me see here. Here actually is hyaluronic acid synthase. But we check the production of different collagens, and we also see this correlation with age. There is a decrease on this production of certain collagen, and sometimes, like when you have treatments that can reverse, you can increase this production again. But there is a decline on collagen, on extracellular matrix in general through aging.
Yeah, there is also an activation of metalloproteinase that degrades collagen faster. So MMP1 is one of the SASP factors that are secreted by senescent cells. And we see an increase with aging, and when we do some interventions, we see a decrease of this enzyme. So the second piece that we mentioned was this algorithm, right? So Steve Harvard, he was the first to design an algorithm that would correlate epigenetic markers with your chronological age. He did this in 2013, and we started using his algorithm, and eventually we saw that the tissue specificity, it really influenced the accuracy of this algorithm. So we built our own algorithm by training with only skin samples, and then we got a better accuracy. So that means that now we can, again, take a piece of your skin and we can measure if your biological age of your skin is like lower or above your chronological age. And this is very interesting because there are several applications for this algorithm. This is just details comparing ours with Harvard in terms of accuracy. But in terms of applications, you can see disease conditions. So cancer or psoriasis, we have shown that it can increase your biological age. Some treatments, we can also validate this. we can validate the efficacy of a treatment by measuring the biological age. And this is, again, the most accurate way to measure because it's objective and you can compare different treatments in the same, kind of using the same ruler. You are not being biased. And the next study that we also want to do, so what they did in this study, and I don't know if you guys have seen that, they met, so 90 patients, they took these 3 drugs for 1 year. This was systemic drugs, not related to skin, but after 1 year, they measured the biological age through their blood, and they saw a decrease in 2.5 years in their biological age, although was not yet significant. But we want to do a similar study with humans that are using these products, and then we can collect after 6 months or 1 year a biopsy of our skin, and we can measure how many years we are rejuvenating. So this is kind of the next steps, because I will show some data here that we have done this in the lab, but the next step is to do this when humans are using our product, and then we can see, because everyone asks how much you can really reverse this age. Can you reverse 10 years? Can you reverse 20 years? We, in fact, don't know. In the lab, you see that the range varies from 3 to 10 years, but in a human setting is a different story. So the last piece of our platform was to design this screening platform to find the new molecules that target senescent cells. And we use the same rationale that the pharma industry uses today to get to a new drug. So we are applying here a very rigorous process to get to a molecule that has been selected to target a specific target. So here we use 2 biomarkers for senescence. One is ATRX, and you can see how different the cells are. For example, an 84 years a cell from an 84-year donor has a different distribution from a cell of a neonatal. The same, you can see the accumulation of senescent cells that are that blue, that are standing blue. So we use this kind of setting, and then we incubate with thousands of compounds until we get to the molecule that can really decrease or reverse these markers.
The ATRX stands for what again? I'm sorry.
ATRX is an enzyme that's required when cells go under senescence. So they form this foci in the nuclei. So these were the numbers of foci that accumulates in the cells. And when the cells are young, you have less foci per cell. When the cells are old, they have more foci per cell.
Thank you.
Mm-hmm. So similar to what was published before, we tested thousands of compounds, and then here in this rectangle in purple, you can see our hits. So these hits are showing molecules that have decreased at least 25% in the level of senescence. That's in this X. And then we do the validation here on different cell donors. And I think it's important to reinforce that everything that we are basing our study and screening, it's human. So we won't have that hurdle of translating from animals to human. And most of the toxicity that eventually is responsible for the failure of the drugs occurs in this transition. So here we are already eliminating all the hits that were already toxic to human cells and to human tissues. So the chances that this molecule will perform better in a human setting are much higher. And there's a chance to apply not only to skin. So skin is it's not only a model to screen molecules for skin, but also to evaluate the toxicity of any molecule that's being developed. So for example, here we tested this drug here, navitoclax, It's one senolytic molecule that we can see that there is a toxicity effect because it's killing more cells than the total number of senescent cells. So it's killing also non-senescent cells. Our molecules, they are up there, so they are decreasing senescence, but they are not decreasing the total number of cells. So this is very, very important when you plan like moving ahead to the next stage, because here you should be very rigorous with what will move forward or not. So we got to OS1 that has shown this ability of decreasing up to 40% the level of senescent cells. We validated at least three different donors, and these are all primary cells. Primary cells are cells that are healthy cells. So again, we are testing this condition that's very similar to the people, to the condition that we want to bring this product, we want to apply this product. This is a very interesting, because you may be asking yourselves how you decrease senescent cells, right? How this peptide works. So one way that we have seen that our peptide is working is by repairing the damage that's accumulating in our cells. So it prevents the accumulation of senescent cells. So in this experiment, we have exposed ourselves to UV radiation, and then here we are increasing the level of senescent cells. You can see here first. And then when we treat, we decrease almost half the number of total senescent cells. So the cell that is treated, it's being more able to repair this damage, and then you have less formation of senescent cells in the end. And the last graphic is also very interesting, because the cell that didn't receive UVB or receive UVB but was treated, the levels are very similar. So again, a very strong effect on DNA repair. And this, hopefully, eventually we can test it for preventing skin cancer, to other diseases that are related to DNA damage, because we know how important, or we also know that DNA damage or UVB damage is one of the main drivers of skin aging.
So where did you find, so OS1 is a peptide, you're saying?
Yeah.
And where did you find it? Where did you discover it? Was it in a library of peptides?
Yes. We started with a library of 200 peptides of one of our collaborators that was working with antimicrobial peptides. From these 200, we got to 4 hits, and then we used an algorithm and we generated more 800 peptides, and then we screened again. So these are all novel sequences, and so when we got to OS1, we could patent, and we have a patent.
And how big a peptide is it? How many amino acids?
It's 10 amino acids.
So small.
Yeah, but now we are testing even a smaller version. We saw a very similar effect with 5 amino acids.
Wow.
Yeah. Yes, this because 10 is an average that can penetrate through your stratum corneum, through your skin, without needing a more sophisticated delivery system. But 5 is even better, right?
Yeah, sure.
Yeah.
So since this was like a library that from the first hits, we generate several new peptides. When we screen this new version of combination, so you combine different ways of having those amino acids, our 5 hits of the second screening, they had the same 5 repetitions, was constant on the top 5. And then we tested this. It's not so effective as the larger one, but has effect as well. So we are trying to optimize to have smaller versions of the peptide.
Amazing.
So now we are combining everything that we showed. So we grow the aged skins. We are treating them with OS1. We also treated them with retinoic acid. That's kind of the gold standard for anti-aging. We are measuring not only how the histology or the morphology of the skin improves, but also the molecular age. So the skin initially had like almost 80 years. With this treatment, we went on average 66 years. And then retinol, you have also a reduction, but you have all the side effects that you can see through that histology, you can see that the layers are coming out. This is kind of a peeling effect that if you have used those acids, you see that your skin will peel off, and it's usually related to inflammation, so usually has a damage that in the long term is not good for you. So what we can do here, we can deliver like a more effective reduction in the skin biological age without any side effects. And we test this also in explants. So these are real skins that we get from, leftover of plastic surgeries. And the skin that is treated, we can see that increase the thickness of the epidermis, this purple area. And we also saw a reduction in the molecular age from 71 to 68. So this is more close to a condition that we would see in vivo because these are real human skins. But there is, and these were treated for 5 days. Again, lab settings is different than a human setting, but already shows the promise of this molecule to be able to promote this rejuvenation. And more than that, we have this ability to test any product that's out there. So a lot of people come to us and they ask us, like, how about this product? Or how we cannot test all of them. But what we were impressed when we start testing some products, if you see that the skin is intact, let me just come here just to show a little better. If you have this layer thicker, you can see that these are good products. When you see a shrinking in the epidermis, like this product, this is basically inducing the differentiation of your skin. So it's kind of killing those cells, and it's forming only like a thicker layer of keratin. This purple is only keratin. So this is not good. The same if you see with other products that are they are full of preservatives. So companies kind of more old, like Estée Lauder, they have, these products, La Mer, that your skin is basically like degenerating here. It's really, really bad. We were like impressed when we saw that. And retinol, it's very classic. Retinol is just inducing this overproliferation, so you lose the upper layers of your skin, but eventually you form it again. So a lot of retinol, it's not good because you can exhaust your tissue, but if you are able to combine with another product that repairs the damage, this could be okay. But it's just interesting because the consumer has no idea, right? When you apply a product, you can't see, unless you have a very bad reaction, you can see that a product like this is bad for your skin. But we can do this, we can show this in a more molecular and cellular level, and then you start like looking the ingredients and you see, it makes sense that's not good for you because the ingredients are not good. So now that we have this molecule and we want to bring it to the market or to consumers, the first thing that we need to evaluate, and very carefully again, is the safety. So we have done all the safety studies to evaluate not only toxicity, but mutagenesis, karyotype, if this would induce any abnormality in your cells or tissue, and we have not found any adverse effect. Then we have done also this sensitization study to see if the peptide could cause any irritation. Everything was clear. And then we moved to a clinical study where we enrolled 22 patients, and we treated them half of their face with a basic formulation, half of their face with the formulation with the peptide. And we saw not only an improvement in the appearance, but we saw an improvement in the skin function. So the first number that we have there is a better skin barrier by on average 15%. So the skin barrier, it's related to the maintenance of water, so you don't have like water loss, and also protects your skin against infections, and even of several diseases are related to when you have like your skin barrier broke. So we saw an improvement, yes, in the appearance, but we also saw an improvement in the health of the skin, in the function of the skin. And now we are thinking bigger. Okay, skin, we validated this application. We are going to bring this to the market. But what else we can apply this peptide for? So we are targeting senescent cells. That's a common mechanism of several age-related disease. So the first experiment that we did to show that this peptide has potential beyond the skin was the C. elegans. C. elegans is a worm that's it's a widely used model to test for aging studies because it lives only 20 days. And then you can test if you are expanding the lifespan of these worms. And now our studies showed that our peptide not only extending on average 21% in the median lifespan, you can see here, but the worms, they were more active throughout their whole life. So that's what we are showing there. Let me show here again. So here in the early days, the control is active, but then it decreases its ability. I have a video that I'll bring for you guys. OS1 keeps the worms active throughout their lives. And this is related to health span, right? We want to be active. We want to be, you know, full of energy throughout our whole lives. And I think this is a very interesting experiment to show the potential of this molecule. Greg, can you show the video for us? So you can see here, there are some that are going pretty fast, like here, here. And even the ones that are moving here, I did it like a circle, but I couldn't keep it. So they move much less, you know. This one, they move much less than those ones. We love seeing them like so so full of it. Yeah.
I always look at Carol. This is me. Like since I started, like I feel so energetic.
So are you guys using it?
Yeah. No, Alessandra has so much energy that I think she's drinking OS1.
I'm not telling what I do in the lab.
Okay, thank you. Yeah, and I think there is a huge opportunity here, obviously, for the consumer market. As a consumer, and as a scientist, I want to be able to bring the benefits of this research earlier to people. Like, we don't want to wait like 10 years so we can have we can feel this research that's being done around, you know, cellular senescence. And if this is safe and we can bring it earlier, we can already create an impact like in people's lives. And hopefully we'll find so much more in terms of helping with preventing other, you know, age-related skin diseases or skin cancer or, you know, psoriasis or rosacea or so on. So very excited about the consumer opportunity, but obviously super interested in the whole longevity market. Very briefly, I don't want to get into the weeds here, but we are going to start bringing this direct to consumer through our website. We are more than only a product. We are this platform of educations, of content around longevity, around skin health. We are very rigorous with all the content, like we curate all the papers and we transform them in, you know, in knowledge for our consumers. We see also a huge opportunity for licensing. A lot of people are interested in OS1, in our platform. We have been approached by several companies. So we see this as an opportunity in the mid and long term. And in the long term, we are also interested in the more therapeutical application for OS1. This could be related to some skin disorders, inflammation, that we have seen a very clear effect to decrease inflammation. But I really hope that we can develop a preventative therapy for longevity. every time that I'm presenting the company, I say that the singularity, I always say that in 2030, we'll have a preventative therapy for longevity. And I do believe that, and I believe that OS1 is a great candidate for this therapy because it's safe and it has a very impressive effect. So another way to differentiate ourselves, we want to call this a supplement and not only a skincare. A topical supplement would make you think that I'm using a supplement because I want to treat my skin. Like you take vitamins, we want to take a supplement for your skin. That's your largest organ and needs to be treated, needs to be nourished. We will also do a subscription model. We also do a subscription model because this is something for the long term. You need to treat your skin. Once you start aging, so that means after you're 25, 30s, you should take care for the rest of your life. So it makes sense. Just a very brief overview of our company since we started at IndieBio in 2016. We went through a few rounds of investment. We raised up to today 7.5, and we are planning to launch our product now in October and then go to Series A really soon. And now the team that's behind all of this that I'm presenting for you guys, the founders, we have this female leadership that I'm so proud of for PhDs. Me, Alessandra, and Juliana, we did our master and PhD in the same lab. We have this expertise, stem cell biology, tissue engineering. Alessandra did her PhD in skin regeneration. And Mariana brings the bioinformatic and genomics expertise to the team. And we were able to bring brilliant scientists from Buck Institute and Stanford. So we are very proud to say that we develop all of this data in-house by ourselves. Amazing. And now we are going to market, so we needed to build a very strong marketing team. And we were very fortunate to find brilliant talent because I think the challenge of marketing is really to translate this science in something that's elegant, that's beautiful, that's cool. And we found this amazing creative designer. If you guys need some, I totally recommend Brian. And we have Dara, that's someone that's super aligned with our vision. So a lot of them very experienced in the marketing, beauty industry, but also with a very strong data and analytics mind. And we have done a lot of collaborations. While we were still doing our own screening, we collaborated with other companies, with other institutions. This helped us not only to grow scientifically, but also explore opportunities to validate other strategies in our platform, in our system. So a lot of them were developing senolytics or reprogramming therapies focused on skin, not necessarily on skin, but other indications. We could be their partners for skin. We didn't move forward except with Otavio. That's the one that we start screening the library because mainly everything that we tested, we didn't see anything that was better than what we have found so far internally. And that's it that I brought for you guys today. Happy to answer any questions.
So let me start by saying what an extraordinary story and congratulations. Just love what you guys are doing. So the question I've been asking all the companies along the way. So where are you in your capitalization? You raised 7.5 million of seed funding so far. Mm-hmm. I heard you say you're going to market this October.
Yeah.
The product becomes available.
Right.
which means you must have some samples you can.
Yes,
Yes,
we
we
do have
You
samples.
can share with us. I saw Maria jump up with joy.
There's a box there full of samples.
That's great. And what you're doing is Series A.
Yes.
When are you looking, and how much do you think that might be?
I think we're looking right after we launch, so probably Q1 of next year, maybe a little earlier. We are looking around 7 to 10.
Another $7 to $10 million
Yeah.
of capital. Great.
Yeah.
What is it that could help you most? What are you looking for to do?
Well, I think in the consumer side now we need more people that have done this before in terms of growth, in terms of brand awareness, and then we are also exploring the other applications for OS1. So we are developing some proof of concepts, and then probably need partners that will take this molecule to the next steps. So,
And do you have any sense of where this oligonucleotide, or I'm sorry, this small peptide works? Is it intranuclear? Have you tracked where it?
Yeah, it doesn't goes to the nuclei. It binds in some receptors in the membrane and also interacts in the cytoplasm of the membrane. And basically, the pathways that we're seeing more regulation, they are related on the oxidative stress pathways, some inflammatory pathways, and gets down to NF-kappa B downregulation, KB downregulation.
Questions? Will, we need to rewrite our beauty chapter.
A little bit, yeah. Well, one thing, we were looking at a company called Proven Skincare, which struck me as slightly similar in that they were using AI and machine learning to look at something like 100,000 different ingredients for skincare products and 20,000 different products. And I was interested in how you compare and how you differ, because it seems like I'm pretty ignorant about skincare, but it seems like, as you can tell, but it seems like there's some similarity in this kind of harnessing of this amazingly powerful new technology to be able to look at enormous numbers of molecules or products or whatever it might be.
yeah, I think Proven, they are using everything that's available and has reviews and that's associated with papers, and trying to combine in different ways, trying to bring this personalization. We come from the angle of more like the aging biology and trying to understand what's the best target, that if we find a molecule that it's more specific, our strategy, in the way that our screening was designed based on a specific target, while Proven is like mixing different ingredients that are already available. It's a classic cosmetic strategy. That's like, let's combine and let's apply to the skin what looks better, we are going to bring to the market. We come from this strategy that's more bottom-up in terms, let's understand aging biology, let's focus on what we believe that's the driver of aging, and by targeting here we'll look better because we are targeting the cause. So one, it's more like a combination of different options and can benefit the skin. They are not validating how it's working as we are doing this more molecular and cellular level. It doesn't mean that's not I didn't test their product, so I can't say if it's not effective. So there are some ingredients in the market that are good. I'm not sure if they are addressing again what we believe that's kind of the cause of the problem, you know.
And I think the main difference is that they use like clinical data, and clinical data like perception. Okay, I use this product, I like or not, this type of information, while we look to molecular and cellular expression or markers like either gene expression or even the epigenetic changes. So we are looking like to markers down to the cellular level, what's changing on the skin when you use any ingredient or any new molecule, while they use more data from, it's a different type of source of data that we are using.
Yeah, we are more kind of the classical, I'd say, drugs, biotech route, and they are, of course, using machine learning, but more based on reviews, ingredients, and I think they also bring some papers, whatever it's described about that ingredient.
Now, you chose not to go and partner with a pharma company here. You chose to go it alone. Did you?
For the consumer, for the therapeutic application, we are looking for not now, but we envision partnering with them to bring this treatment to the market, yeah.
And have you looked for any adjacent cell differentiations like tendon or ligament or anything that's related?
Yeah, we're planning on look a little bit to osteoarthritis because everything we see a lot of decrease on inflammation, and we have data that our peptide modulates a little bit of inflammation. So diseases that has like inflammation as a cause of the disease, a high inflammation, we are starting to like try to do small experiments of proof of concept to try to identify which one would be more promising that we could move forward or try to find the right partners for that.
Yeah, I want to apply on my knee because my knee is not so good. Just kidding, but yeah, I do believe that OS1 has this very promising role in decreasing inflammation, and eventually we could have something systemically. It would be like ideal, but probably starting like local or some. Mm-hmm.
Questions?
You indicated on the commercial side that it's topical, correct?
Mm-hmm. Correct.
Does that mean it's effective at the point of contact, or is the idea, for example, I can't wear anything on my face? So as much as my face needs it, anything I put on my face, it not only does not help, I have rejection.
Mm-hmm.
So ideally, if this is all topical, it's not the only area of my body that I need assistance with.
Yeah.
Am I then going to be thinking the lotion or
Mm-hmm.
whatever source it is needs to go full body, and it then hits those particular, like I said, point of contact?
Yeah, so initially the treatment is going to be for the skin. So we are going to start only with a face product. Eventually, we do see that makes so much sense to bring a body product because after 50s, your skin really starts like needing this repair. If you need something that's more like a treatment, if you, for example, if you are targeting your knee or some kind of joints, then it needs to be injected. We don't believe that the penetration through the skin would get to a point that needs, you know. And eventually, we even think on some applications, for example, combining this with collagen and hyaluronic acid, this could be an application that you could do through the dermatologist, you know, since you can't handle any. So there is this injectable market that is also an option to explore, combining with this kind of extracellular matrix that has been used, but then something that a molecule that has this functional activity.
Oh, great. Thank you.
Of course.
Other questions? So, October, direct to consumer.
Mm-hmm.
I'm happy to help promote you, so please, I'll get your contact details. The capital you raised in your Series A is going to be used for different products or just to grow that product line?
Initially, we'll focus on this product line, but we'll feel from the consumers. So if we are seeing that we can have impact in other areas, we are going to focus on expanding as well. But the projections that we have right now and this raise would be to reach like break even in 2-3 years, would be only focused on this product. We are not counting any other source of revenue, even licensing other things that we are exploring already in the midterm.
Fantastic. And one last question for me. I saw the measure, the study you did was a reduction in skin age from like 76 to 61. Have you looked, is that where the steepest part of the curve is? Have you looked at, like, if someone who's got skin at 45, if that reduces it down?
So this is a very kind of expensive study, so we usually do it like, we don't do like every day. So that skin was 79, right? Yeah. The one that we did. Okay. So what we are planning now is this clinical study that we are going to enroll 12 patients, and then we want to enroll ourselves. So we want to see.
And it will be open to others to enroll.
So we want to see if in 6 months treating with the product, how much we can reduce in a younger age. So right now we used like older ages to try to see a more significant effect, but we'll have.
And it's application once a day?
Yeah.
Twice a day,
Twice
in
a day.
the morning and the evening.
You put it in the bath and you get in the bath. And
It takes 2 minutes to apply.
All right, any other questions, folks? One last question, Michael.
I might have asked you this before, but in terms of, so you talked about subscription model, but since it's killing senescent cells, why not just apply once and wait a year again to apply? Why regularly? Is it just a concentration? Are you trying to control, like, or is it just depend, you need the extra time?
Yeah, so the evidence that we have right now is that we are preventing the accumulation of senescent cell much more than inducing apoptosis. If you are inducing apoptosis, it's in a very lower rate that it's still okay if you are using every day. So apoptosis we see only when you have like high stress, an acute stress.
Can you define apoptosis for everybody?
Okay, so apoptosis is cell death. So the way that these molecules that work targeting senescent cells, they can both eliminate, induce the cell to die. These are the classic senolytics. Or they can prevent those cells to become senescent in the first place. And we see that our molecules work more expressively in preventing the formation because of this DNA damage repair. So let's say one cell is like early stage senescence. The cell doesn't go to full senescence because we are able to prevent, this evolution of the damage.
Yeah. And just complementing, the skin, we are exposed to damage, especially like UV damage all the time. And all this damage that we are exposure are inducing new senescent cells to be formed. So we are more like preventing this new formation than just like one application, you eliminate whatever you have, but then you go to the beach, stay like 10 hours in the sun, and then you can't like, so we see a very nice repair on those very stressful situations, especially through UV radiation. And we even think that this approach is more interesting for treating the skin than a very strong senolytic that I would pass now will kill all my senescent cells or most of them, but I would be exposed to damage all the time and I would just like create new senescent cells. So we have this like continuous application.
Well, we wish you great success, for sure. So if you please give it up for OneSkin.